Good day to all watching the video. This video is on urea recycling. That is the role of urea in building a hyperosmotic medullary interstitium. Please remember, urea contributes 50% to the hyperosmotic medullary interstitium. Urea is a waste product of the body, but it is a permeate solute, means that it easily passes through the cell membrane, and as a result it can accumulate in the cells of the body. Hence the plasma has to recycle several times to rid the body of the toxic urea which is produced. Otherwise it will accumulate in the body and lead to great toxicity. Whenever we discuss any concept, we discuss at a structural and functional level, that is at the level of the nephron. The nephron is the structural unit of the kidney, but it is also the functional unit. That is, all the functions which occur in the kidney can take place in one nephron. There are a million nephrons in each of the two kidneys. Urea is almost freely filtered as freely as water. So these are the efferent and the efferent arteriole, and these leading to the vasa recta. So you guessed it right. This is the juxtamedullary nephron. The juxtamedullary nephron have vasa recta, while the cortical nephrons, which make up 5/6 of the nephrons or 88% of the nephrons, have peritubular capillaries. The renal circulation is a portal circulation with a glomerular capillaries and a peritubular capillaries. While the juxtamedullary nephron have vasa recta instead of peritubular capillaries. These vasa recta have a big role in maintaining the hyperosmotic medullary interstitium. We'll label each of the parts. The proximal convoluted tubule. It is convoluted at some parts. In some parts, then there is a loop of Henle which has a thin descending limb. But in this case, it has a thin ascending limb and a thick ascending limb. TAL is thick ascending limb. Then there is the early distal tubule. If there is an early distal tubule, there is a late distal tubule which is a functional unit with the collecting duct. Large number of late distal tubules form one collecting duct, and the collecting duct plays a critical role in urea recycling. So there is a thin ascending limb, and the thin ascending limb also acts as a functional unit. We have seen that the thin descending limb and the thick ascending limb play a role in the countercurrent processes, while the thin ascending limb has a role in the handling of urea. Now when urea is filtered, and 100% is there in the initial part of the proximal tubule. But the proximal tubule will reabsorb 50% of the filtered urea. 50%. It will reabsorb. Reabsorb because absorption occurs in the intestine when it is filtered by the nephron, and then in the tubules it is reabsorbed. It is called reabsorbed. This then goes This then remains as 50% at the end of the proximal tubule. So at the end of the proximal tubule, only 50% of the filtered urea remains. 50% is reabsorbed in the proximal tubule. So at the end, only 50% remains. This is the first step. The second step is very interesting. There is secretion of urea. Secretion means passing from the capillaries into the tubular lumen. Absorption is passing from the tubular lumen into the capillaries, while secretion is passing from the capillaries into the tubular lumen. Here from the peritubular capillaries into the tubular lumen. And 50% is secreted. So again, at the beginning of the thick ascending limb, it is 100%. The urea concentration at the end of the thick ascending limb and at the beginning of the proximal tubule, at the beginning of both these segments, is 100%. In the thick ascending limb, 30% is reabsorbed. 30% is reabsorbed. So we have the third step. So when it passes through the early distal tubule and late distal tubule and enters into the collecting duct, and the collecting duct, there is 70% of the urea remains. 70% of the urea remains. There's a large amount of urea. And this enables the recycling. %70 of the urea remains, which enables the recycling. Hence, 55% is then reabsorbed through the inner medullary collecting duct. The collecting duct also has two parts, outer medullary collecting duct and inner medullary collecting duct. And it is through the inner medullary collecting duct that 55% of the urea is reabsorbed, out of which 50% goes to the thin ascending limb through urea transporter UTA2 into the thin ascending limb, and then 5% goes to the vasa recta to be recycled. This is how urea is handled by the nephron. And this recycling of urea then contributes to the buildup of solute in the medullary interstitium. Solute builds up. What are the solutes made up of? Sodium and associated anions. So sodium, chloride and bicarbonate. Then 4% is by glucose and urea, and the rest is by plasma proteins. This is the medullary interstitium where urea is built up. Please remember that the hyperosmotic medullary interstitium is not allowed to be lost by the counter current exchange mechanism. Building up is by the counter current multiplier mechanism and the medullary interstitium. Urea contributes 50% of the total osmolarity in the medullary interstitium. So at the end of the proximal tubule, there is 50% of the urea, 50% is secreted in the thin ascending limb. At the end of the in the thick ascending limb, 30% is reabsorbed, 70% remains in the collecting duct, out of which 55% is reabsorbed, and thus it is recycled. So this is the story of urea in the nephron. Thanks a lot for watching my video. Please like and subscribe to my channel. Enjoy the video. Please remember that urea is a very important contributor to the maintenance of to the buildup of solutes in the medullary interstitium. So we have the countercurrent multiplier mechanism which builds up solutes in the medullary interstitium, the countercurrent exchange system which prevents the solutes from being lost. We have urea which builds up solutes in the medullary interstitium, and then finally we have the countercurrent exchange system which prevents it from being lost. Thank you for watching my channel. Have a good day and enjoy the day.